we are going to see the dihybrid cross. So, dihybrid cross, Mendel in his experiment, he was taking a pea plant. It has two pairs of contrasting traits. So, two pairs of contrasting traits he was focusing. So, he was focusing on seed shape and seed color. So, what are the parents? Actually, he has selected the round shape and green color seed. So, with that plant, pea plant, he crossed with wrinkled seed shape and yellow color. So these are the two pairs of contrasting character he was taking as a parents, and he was cross pollinating. So he cross pollinated these two plants, and one is male and another one is female. So I am writing the phenotype of parents. Phenotype of parents, only physical appearance we have to write. So round, green, wrinkled yellow or phenotype of parents. Then genotype of parents. Genotype of parent means it is we are writing a gene combination. So when we are writing a gene combination, we have to write whatever it is dominant character, we have to write with capital letter. So here round is dominant. So round is dominant. So we are writing with capital letter, capital R, capital R. The corresponding recessive character, it is wrinkled. So wrinkled, we are writing with small r, small r. So the same way, yellow. Yellow is dominant. So yellow is dominant. So I am writing the yellow with capital letter, capital Y, capital Y. And the wrinkled, it is corresponding recessive, it is green. Corresponding recessive, it is green. Green, I'm writing with small y, small y. So this is the way we are writing a genotype of parents. So now when we cross-pollinate this two pea plant during gamete formation, so how the formation of gamete it takes place. Gametes formation, it is in certain order the gametes are produced. First, we are going to take the R and join with this Y, and then R, the same R with this Y. So first let me this R, capital R with small y. So I am writing the gamete, and then again the same R I am taking and joining with this small y. So I am writing the gene combination capital R, small y. Now the next chance it is this capital R. So I am taking this capital R joining with this y. So the gamete will be carrying capital R, small y. Same this R I am joining with this small y. So this is the probability of gamete formation. So if you follow this, you are able to write the gametes easily. So these are the four types of gametes you are getting. But fortunately, all the four are same type. So all the gametes, it is they carry capital R and small y. So gamete formation you have to know very clearly, because you will get in board question, case study question, you will they will ask you to write the gamete formation. So now for one male parent, you have formed the gametes. So this we can say all pollens. So as it is a male plant, all are pollens, gametes, male gametes. So now you can form the gamete for the female plant. Female plant, how is it producing a gametes? So I'm taking a small r, this r with this capital Y, small r with capital Y. The same R is joining with another Y. It is capital Y. Now I am taking the next R and another capital Y. It is or Y. So the same way, the small r, I'm taking the small r with this Y. So this is the way four gametes are produced. But all the gametes are same, similar, because all carries small r, capital Y. But this gamete formation, how they are forming a gamete, what are the chance of forming a gamete? So this you have to learn carefully because you will get in board exam. Okay, as it is a same gamete here, same gene combination is present in the gamete, it is easy to write, but other cases it will be difficult, so you have to know. So these are the gametes it's formed. So these gametes are female gametes, so we can write it as egg. Female gametes or egg. So what you will get in F1 generation, F1 generation or progeny or filial generation, we will get all will be round and yellow. Round when these two are joining. When these two are joining, as it is same type of gametes, you will get only one possibility, capital R, small r, capital Y, small y. So all will be round yellow. So phenotype of F1 generation, it will be round yellow. Phenotype means physical appearance. So round yellow is a phenotype. So what is the phenotype of F1 generation of dihybrid cross means you have to write round yellow. What is the genotype means you have to write capital R, capital Y, small y. This is the genotype. So now what he is doing, he is self-pollinating F1 generation. So when he is self-pollinating F1 generation, he's when he is self-pollinating F1 generation, he has to cross this two plants, F1 plants. So I am writing capital R, small. Self-pollinated with another F1 plant. So these are the parents now. F1 generation is self-pollinated. So I have written a male plant and female plant. So during gamete formation, so F1 generation is producing a gametes. So what are the ways in which you have to you have to write the gametes? I taught you already. I think you are able to write clearly now. Now I am taking a same R joining with this Y. So gamete will be R, capital Y. Same R, capital R. I am joining with another Y. It is capital R, Y. Now it is I am taking a small r and capital Y. So I am getting a gamete like small r, capital Y. And the same small r I am taking and joining with this small y. So the gamete will be carrying small r, small y. So I get the four different types of gametes. So this gamete formation, you have to know, because in the board they will give like this one genotype and they will tell you to write the gametes. So next, the same way you will get the gamete formation in this side also. So when you are joining the same way, when you are joining the capital R with this Y, this is the way how the formation of gamete it takes place in the male plant. The same way there will be four different gametes are produced in the from the female plant also. So now all the gametes you have written. So it is easy to write the individuals by drawing Punnett square. So for F1 generation, what are the combinations you will get that plants? What are the gene combination it will be produced? You can draw a Punnett square and you can write all the male gametes. So whatever the gametes it is produced, whatever gametes it is produced here, four different types of gametes that I am writing in one side of the Punnett square. It is capital R, capital Y, capital R, small r, capital Y, small r, small y. So first we are writing a male gametes here and female gametes we are writing here, this side. So it is easy to write the gene combination. So seeing this two, capital R, capital Y. So this column and this row and column we can see and write the gene combination very easily. Capital Y, small y. So this is the way we can see the column and row and write the combination very easily. So we are getting a 16 individuals. So wherever you have a dominant character, only the dominant it will be expressed. So you can say it is round yellow because both are dominant. So this is the way round yellow. Here also round yellow, round yellow, round green, round yellow, round green, round yellow, round yellow, wrinkled yellow, wrinkled yellow, wrinkled yellow, round yellow, round green, wrinkled yellow, wrinkled green. So these are the ways you will get 18 individuals, though these 18, sorry, 16 individuals. So these 16 plants, F2 generation, produced in F2 generation. So F2 generation, if you are seeing this, 9 plants are round yellow. 9 plants round yellow. Round yellow, dominant character. 3 plants, it will be round green, having the phenotype of round green. 3 plant, it will be wrinkled yellow, and 1 plant is wrinkled green. In this gene combination, what are the parental combination? Wrinkled green. So parental combination, round green is parental combination. So similar to parent, and wrinkled yellow, it is similar to parent. You can see the parent. You can see the parent, round green, wrinkled yellow. So parental combination here it is round green and wrinkled yellow. So this round yellow, it is a new combination. new combination or recombination. So here wrinkled green is new combination. So in a dihybrid cross, two new combinations are produced. So two new combinations are produced. So we are able to prove the second law. So what is the second law? Actually, the Mendel's second law of independent inheritance. So law of independent inheritance. Law of independent inheritance. Inheritance proved with the help of this dihybrid cross. Actually, the law of independent inheritance or law of independent assortment. It states that inheritance of one character is independent that of another character. So inheritance of one character, the round yellow, it will be independent that of inheritance of other character like wrinkled green. So this is what proved in this dihybrid cross. So in the dihybrid cross, the law of independent inheritance or the law of independent assortment. This law is proved because in the F2 generation, the two new combination plants are produced. So it proves that the seed shape and seed shape and seed color separated independently during gamete formation. Separated independently during gamete formation. So it proves that the law of independent inheritance by Briher John Mendel. Phenotypic ratio of F2 generation in dihybrid cross. Phenotypic ratio of F2 generation in dihybrid cross is 9 is to 3 is to 3 is to 1. If you take the genotypic ratio means it is gene combination. You are getting a gene combination 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So total you can see the 16 gene combination 3, 4, 5, 6, 10, 16. So 16 types of gene combination you are able to see. You can see the column. So gene combination, it will be this one. So you can see the gene combination. It will be differing. This is one different round yellow, wrinkled green. It is a different round yellow, round yellow. This is the way you are getting, and you are getting round yellow, round green, wrinkled yellow, wrinkled green, round green. So these are the so many type of genotypic ratio you are getting. Genotypic genotypic individuals or combination you are getting. So the genotypic ratio is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So ratio means you have to write the number, and simply genotype means you have to write the gene combination alone.